Yes, students. In our last class, we saw how biotechnology has provided us the way to protect the crops from different types of pests, insects, etc. Biotechnology also finds its major use in the field of medicines. We all know a huge population of our world suffers from the disease diabetes mellitus. It is one of the major cause of a large number of deaths that occur across the globe. Pancreas gland, which is a mixotrophic gland, has an endocrine and an exocrine part. The endocrine part of the pancreas synthesizes the hormone insulin, which maintains the balance between the glucose and glycogen. Insulin hormone, as you know, increases the permeability of the cells to take up glucose. It also stimulates the liver cells to store glucose and convert it into glycogen. But if the pancreas fails to produce enough amount of insulin, the blood glucose level rises as the cells can no longer take up the glucose and decrease the blood glucose level. Now, to decrease this, initially some medicines are given to stimulate the pancreas to produce more of insulin. But if this fails, then the insulin is needed to be provided from outside in the form of injections. It is not given orally because it is a protein hormone and it will be digested in the stomach. So, insulin cannot be given orally. It has to be taken in the form of intradermal injections so that it is absorbed directly into the blood. This insulin initially was extracted from the bovine animals, the cattle, the pigs. They were slaughtered, their pancreas were collected and the insulin was extracted from there. This was not only a very expensive method, but also it caused a number of immune reactions in various number of patients. So, biotechnology came up with the synthesis of artificial insulin, which is exactly like the human insulin because it came up with the synthesis of insulin from the human genes. The genes for insulin synthesis were isolated from the leukocytes and then they were introduced in plasmids. So what was done? Actually, human leukocytes were taken and the insulin producing genes were isolated. But before this, I need to tell you one thing, that the insulin is actually secreted in the body in the form of proinsulin, which has 3 peptides. Peptide A, peptide B, joined by a short segment of peptide C. And as I said, when it is secreted, it is secreted as proinsulin. Then it undergoes some changes to form the active insulin, which is a dipeptide consisting of only A and B peptide. The C peptide is eliminated. So, the leukocytes were used to extract the genes for this insulin A chain polypeptide and insulin B polypeptide. So, these genes were isolated and were inserted in a specific plasmid, PBR322. which has, as you all know, a promoter gene, a beta galactosidase gene, and the A gene is introduced in it. Of course, you have an antibiotic resistance gene to find out the efficiency of the ligation of A gene. Similarly, another plasmid is synthesized with the B gene. Promoter gene, beta galactosidase gene, and the B gene, or the beta or the B peptide gene of insulin. So these two artificial plasmids are synthesized separately. Then they are introduced in the Escherichia coli, which is plasmid free. So in plasmid free Escherichia coli or E. coli, these synthesized or recombinant RNA are introduced and they are made to replicate. They increase their number and then the products are extracted from them. So the product that we get from these is a beta galactosidase, insulin A peptide, beta galactoside, insulin A peptide, beta galactoside, insulin B peptide. Then they are treated with cyanogen bromide to separate the beta galactoside from the A gene and the B gene respectively. Then the A gene and B gene are made to combine with each other in the bioreactor. And then synthesized insulin, which is identical to human insulin, called the humulin, is extracted or isolated from the bioreactor. In this way, we can synthesize huge amount of humulin in a short span of time, and that also exactly having the amino acid sequence of the human insulin. So it will not cause any immunogenic reactions. Totally safe, as well as they are cheap in the market. So this made available huge amount of human insulin in the market for the patients with diabetes mellitus. So a huge boon in the field of medicines. Similarly, nowadays very many medicines are synthesized in this way. The gene therapy is introduced in the patients with gene problems, inherited gene defects. At the same time, the gene therapy has made possible to introduce the genes for that is lacking in the patients with hemophilia. This process is ongoing to control thalassemia in the same way. And maybe in future we have the answer to majority of the diseases in the inherited diseases in the form of gene therapy.